Dear students, Assalamu alaikum. Today we are going to talk about our course Molecular Biology, course code BCHM 221. This is lecture number 12 of the series of our online lectures. Today we are going to talk about translation in prokaryotes, elongation and termination. In the previous lecture we talked about initiation of the process of translation. Now we will move towards the processes of elongation and termination of translation in prokaryotes. So as I mentioned in the previous lecture, we are going to talk about we are going to talk about this process of translation in a stepwise manner, because this is a vast topic, so it would be helpful if we understand it in a stepwise manner. So I hope you now understand the process of initiation, now elongation and termination. The outline for today's lecture is we will talk about the elongation of translation. Then we will talk about amino acid transfer RNA delivery. Then we will talk about peptide bond formation. Remember that in the process of translation, there are information are provided by the messenger RNA in the form of codon, but then the different amino acids, they are linked together by peptide bond in the newly formed chain of amino acids. These amino acids are linked together by the peptide bond. So we will talk about how the peptide bond is formed. Then we will talk about a very important step, that is translocation. Translocation happens when amino acid is added, and then the translation machinery moves towards the other codon and incorporate or add further amino acids. So we will talk about this process of translocation, and then finally we will talk about the termination, how this process of translation is terminated, or it comes to its end or conclusion. So this is the outline. Now we move towards our lecture, elongation of translation. Now, with the formation of 70S initiation complex, as you saw in the previous lecture, the elongation cycle can begin. Once the 70S initiation complex is formed, elongation cycle can begin. It involves 3 elongation factors: elongation factor Tu, elongation factor Ts, and elongation factor G. And then the other factors that are required is GTP, guanosine triphosphate. When it is hydrolyzed, it work as a source of energy. Transfer RNA and the 70S initiation complex. So these are the different factors which play a role in the process of elongation of translation. Now, elongation, the process of elongation is divided into 3 steps. The first among these 3 steps is amino acid transfer RNA delivery. Amino acid transfer RNA delivery is important because elongation factor Tu is required to deliver the amino acid transfer RNA to A site and energy is consumed in this process, in this step, by hydrolysis of GTP. The released elongation factor TU GDP, because once the GTP is hydrolyzed, it is converted to GDP, guanosine triphosphate to guanosine diphosphate. Complex is regenerated with the help of elongation factor TS. In the elongation factor TU, elongation factor TS exchange cycle, elongation factor TS displaces the GDP and replaces itself by GTP. The resultant elongation factor TU GTP complex is now available to bind another amino acyl transfer RNA and deliver it to ribosome. All amino acyl transfer RNA can form this complex with EFTU except the initiator transfer RNA. Another step in the process of elongation is the peptide bond formation. Like I told you before, that during the chain of during the process of translation, multiple amino acids, they are added to the chain, and these amino acids are linked together by a bond that is called peptide bond. And now during the elongation, peptide bond formation takes place. After amino acyl transfer RNA delivery, the A and P sites are both occupied, and the two amino acids that are to be joined are close to each other. The peptidyl transferase activity of the 50S subunit can now form a peptide bond between the two amino acids. So the peptidyl transferase activity of the 50S subunit, it help in to form a peptide bond between the two amino acids. Now the step number 3 of the process of elongation is the process of translocation. A complex of elongation factor G, translocase, and GTP binds to the ribosome and is an energy consuming step. The discharged transfer RNA is ejected from the P site. The peptidyl transfer RNA is moved from A site to P site. The messenger RNA moves by one codon relative to one codon to the ribosome. GTP, after the hydrolysis of GTP, GDP and EFG are released. A new codon is now present in the vacant site. Now another look at how the process of elongation of translation takes place. This elongation factors that are involved are elongation factor TU, elongation factor G, elongation factor TS, all GTPase proteins. Now how this process occur in a stepwise manner. Elongation factor TU attached to amino acyl transfer RNA. After this, transfer RNA attached to A site elongation factor Tu bind to elongation factor Tu binding site of ribosome. After this, elongation factor Tu hydrolyzes GTP to GDP and inorganic phosphate. Codon-anticodon bond is formed. Elongation factor Tu fall off carrying GDP. This is followed by elongation factor Ts hydrolyzes its GTP to release energy to form bond between GDP and API and make GTP again in the elongation factor Tu that fell off. This is the diagrammatic representation of the process of elongation. You can see here is the messenger RNA codon degradation. Here is the P site, here is the A site, here is the E site. So here is the P site transfer RNA. This is the newly formed peptide. Now you see here certain elongation factor Tu, GTP and amino acid transfer RNA ternary complex. It is added here. You can see in the next step the process occur as I discussed in the text. This is the E site, P site and A site. This is the 5' to 3' messenger RNA. This is the complex that has formed and now in the next step some of these factors as we discussed in the text, they are released. The elongation factor Ts again enter the system. These different factors, they are recycled and they again come in this shape EFTU, GTP, AA and transfer RNA, and they will again help our work in the process of adding another amino acid to the sequence. Here is the broader sequence. This is the messenger RNA 5' to 3'. These are the codon. This is the transfer RNA. This is the codon. This is the anticodon. This is again the codon. This is the anticodon. This is how this bond formation is taking place, is shown in the yellow. So how the peptide bond is formed, and peptidyl transferase has an important role in the oral transfer. And now we talk about the translocation. The machinery, the messenger RNA, this is how the translocation, it is read in a 5' to 3' direction. This is the E site, this is the transfer RNA that is attached. This is the P site, this is the A site, this is the serine, and this is the methionine, the starting amino acid. The peptide bond is formed between the newly formed amino acids, and or the process of translocation takes place. Now we move towards like this process of elongation continues. Overall, this process of translation, elongation of translation, it continues for period of time until and unless stop codon is reached, because now for different proteins we have different coding messenger RNA, so it depends on how many codons are present in the messenger RNA. This process will continue, uh, until and unless a situation is reached where where the translation machinery encounters the stop codon. Once the translation machinery encounters the stop codon, then this will be the beginning of the end, or the process of termination will start. Termination of translation happens when the A site of the ribosome faces a stop codon, UUA, UGA, or UAG. Sorry, one is UAG and the other one is UGA. This is a mistake. Now, remember from the lecture of genetic code, we talked about that there are 64 possible codons. Among those 64 possible codons, we have 61 base codons which code for amino acids, and there are 3 codons, UA, UGA, and UAG, that are stop codons. When the translation machinery encounters the stop codon, no further amino acids can be added. It signals to the system that the translation machinery should disassemble, and the process of translation, or the process of formation of the newly formed chain of amino acid has to stop. So when this happens, no transfer RNA can recognize it, but a releasing factor can recognize the stop codons and causes the release of polypeptide chain. In prokaryotes, once a stop codon occupies the A site, 3 termination or release factors, RF1, release factor 1, release factor 2, and release factor 3, contribute to the hydrolysis of peptide transfer RNA bond. So, at the moment the system encounters any of the stop codon, the A site 3 termination or release factors RF1, RF2, and RF3 contribute to the hydrolysis of peptide transfer RNA bond. Release the free polypeptide and last uncharged transfer RNA from P site. The dissociation of the 70S into 30S and 50S subunits takes place. Now the ribosomal machinery dissociates into its components, that is 30S and 50S subunits. Release factor 1 binds A site and release the polypeptide and uncharged transfer RNA. Release factor 2 releases the release factor 1 from A site and release itself as well from translation binding site present on large subunit. RF3, release factor 3 function is unknown. Another factor called ribosomal releasing factor causes the dissociation of 70S complex. So this is how the diagrammatically the process looks like. This is the newly formed chain. Here is the system encounters stop codon UAG, UAA, or UGA. Now this is the result of encountering the stop codon. The free polypeptide is released. GTP is hydrolyzed to GTP or hydrolyzed to GDP. The ribosomal machinery, this is the messenger RNA 5' to 3'. This is the 30S subunit, and this is the 50s subunit. They are disassembled, and now overall the process of translation in prokaryotes comes to its end. So this is the free polypeptide that has been formed. The information was provided initially by the DNA, which was transcribed to messenger RNA, and then now the messenger RNA, working as intermediate, provided the information for the production of this free polypeptide. This free polypeptide is then further processed before it is used. It is used through it is processed through post-translational modifications, or PTMs. We will talk about this in the coming lectures, but this is how the process of translation comes to end. Thank you for watching, and if you have any questions, please feel free to contact me. I'll be happy to answer. Thank you.